from the Fundació IMIN. She's going to talk about functional and structural characterization of a new human ultrapotent pan-neutralizing SARS-CoV-2 monoclonal antibody.
No? So, is this one? Okay. No. No. No, sí, que tienes la presentación ya. La traí ayer. Vale.
I think her talk is not ready. We can move to the next one.
Yep.
Yeah. Okay. So the next speaker is Mario Vázquez from the IDIBAPS, and he's going to talk about development of a CAR T-cell therapy for SARS-CoV-2.
Hello everyone, I'm Mario from Manel Juan's lab, and I'm going to talk about my research in the lab. I'm going to talk about SARS-CoV-2, but I think that since we have already heard a bunch of presentations in the past few years about this virus, I'm going to skip the main introduction and get straight to the point. We have seen in the literature that there are certain subgroups of the population that still have a poor outcome from COVID-19 from COVID-19 disease. Some examples are CAR T-cell therapy patients that have B-cell aplasia, and also kidney transplant individuals that cannot benefit from vaccination as they are not able to induce a proper immunity response. So since these patients also, well, it was described that in these patients there's also a prolonged viral replication, which in this article they say that as it is prolonged, the virus has a higher or more time to accumulate more mutations, and they say that it is possible that these individuals are a niche for the development of new strains that could possibly escape the immunity that we have acquired with the vaccines. So they say that there's the need to develop antiviral strategies in order to first cure these individuals and protect the global population from new possible variants. So the answer that we have in the lab for that is the generation of a CAR T-cell therapy. We had two antibodies, C8 and C10, that were directed against the RBD of the spike protein. And well, we combined them with either CD28 or 4-1BB costimulatory domain. We generated the lentiviral vectors and transduced them in healthy T cells isolated from peripheral blood. And well, we evaluated CAR expression on T cell surface by flow cytometry, and as you can see, we were able to detect the CAR in all four constructs. Next step was to see if the transduction of this transgene was affecting T cell fitness, and for that we did an expansion protocol activating T cells with anti-CD3 or anti-CD28 molecules, and after 10-day expansion check if they reach the threshold of 5 population doublings. We saw that all of the CAR Ts except the C8BBZ1 were capable of proliferating properly as the untransduced control cells. Okay, then we had our CAR Ts, but as in the lab we don't have a biosafety level 3 facility, we weren't able to work with the real virus, so we needed to think something to obtain an in vitro model to work. Then we designed a transgene that codified for the spike protein of the original strain of the virus and transduced two different cell line models, HEC and A549. We were able to detect the surface expression of the spike protein, and we obtained, well, I don't know if by chance, but high expression cell line with the HEC cell line and mid-low expression with the A549. Well, then we needed to do the science, and we did the most simple experiment that we could have done, which is a simple co-culture of target cells and T cells to see if our CAR-Ts were able to recognize the target cells and activate. After 24 hours of co-culture, we collected supernatant and analyzed interferon gamma production and interleukin-2 via ELISA. As you can see, the C10-derived CAR-T cells were capable of activating as they generated interferon gamma and interleukin-2. Okay, next step was to see if our CAR-Ts were capable of killing, and for that we used the RTCA excisions machine, which is a machine that is capable of analyzing how many cells do you have in a well. And, well, I'm going to explain it with the actual graph. It's a real-time fashion machine, so the idea is that cells start to grow, so you're going to see an increase in cell index over time, but if you add a toxic compound, or in this case, a CAR-T cell, you're going to see a decrease in the proliferation rate, or if the cytotoxicity effect is high, you're going to see a decrease in the total cell index. So in this case, the graph talks by itself, and we were able to, well, our C10-derived CAR-T cells on the C8 one were able to kill the in vitro infection model. We also needed to check if that was due to, well, if that cytotoxic effect was specific, and we did the same experiment, but with target cells that were not expressing the spike protein, and we could not see any cytotoxic effect. Here are the group results for the 293 model and the group results for the I549. In all cases, in the end, we obtained that the C10-derived CAR-T cells were the one exerting a higher cytotoxicity. Okay, so we wanted to know a little bit more about CAR-Ts, and for that, we did an in vitro persistence assay. It's pretty much the same as before, but this time we are challenging our CAR-Ts to more and more cells over time to see if, in the end, they are able to proliferate and maintain their cytotoxic capacity. Well, results pretty much were what we expected. C10-derived CAR-T cells were the ones that proliferated more during several challenge cycles, and also were the ones to maintain their cytotoxic capacity. Okay, and we were happy because it was pretty much working, but we were concerned because expressing the spike with a transgene was not probably the physiological condition that we were gonna find in the real situation. And well, we did ask for help, and we did this same assay, but this time using infected cells with the real virus in a biosafety level 3 facility. This was done by our collaborators in Montpellier. And well, the idea is the same as before. T cells, target cells were seated in the plate. After 60 hours, we infected them with the virus. And then 20 hours past the infection, we seeded the CAR-Ts. And we waited for the result. This is the result, and let me explain it a little bit. Viruses have a cytopathic effect, so we needed to add a control, because the virus also kills, in red, also kills the infected, the cells. So, well, in the end, this condition allows us to establish a threshold that allows us to differentiate between the cytotoxic effect that was due to the infection or the CAR-Ts. So in all of our infected conditions, our CAR Ts were able to diminish the cell index, cell index lower than the threshold. So we think that our CAR Ts are capable of recognizing the real infected cells. So that's pretty good news. And well, as you know, we have been doing these experiments for the original strain, but over time there have been appearing new variants, Delta, Omicron, I don't know how many, a lot. So we decided to see if our CAR Ts were still capable of recognizing the virus that we have nowadays, and we did affinity studies for the Delta and Omicron strains, which were at the moment the ones more abundant in circulation. Well, what we saw is that our antibodies were capable of maintaining their affinity within an acceptable range. So we think that they are still going to be capable of recognizing the strains that we have right now and probably new strains that may sprout in the future. And well, to conclude, we think that generation of CAR T-cell therapy for viral infections is possible, not only for SARS-CoV-2, but for other types of viruses. Then, more in specific, that our C10-derived CAR T-cells can recognize the spike on the infected cells and in our model in vitro, and that these CAR Ts are going to be still capable of recognizing new strains that may sprout in the future. And well, last, to thank all of you for your attention and all of my lab members for their help every day. Thank you very much.
Thank you. We have a question here.
Yes, thank you. Do you have any information on which is the epitope that is recognizing this antibody and
the epitope? I know it's directed against the receptor binding domain, but I don't know more specific because the antibodies were designed by another lab of a consortium that we are from, and I don't really remember which epitope it is. I'm sorry.
Okay, thank you.
Can you hear me? Yes. So the thing is that, as you know, probably there is a secondary effect of CAR T-cell therapy, which is cytokine storm, which is the same what happens in severe SARS-CoV-2 infected people. So at what type of patients could it be administered this therapy, this potential therapy?
Yeah, well, this is something we are still debating with Manel and other of our supervisors. But we think that instead of administering it once the patient is already with severe COVID, we want to administer it as a prophylactic therapy, so they have a protection before they are even infected, which is something that, well, needs a little bit more of research because CAR T-cell therapy is real expensive. But we are also working on that in the lab in order to lower the costs of that kind of therapy.
Nice work, Mario. So in the same way, do you think that, if I'm not wrong, we need to prepare the patient with some chemotherapies before the infusion of the CAR T cells, that this will be able to do it before the infection, or it will be like something that we must think a lot?
I think I don't get your question at all. So you're saying that we need to condition the
I'm saying
patient?
To infuse the CAR T cell, we have to condition, or make a condition in the patient,
Yeah.
like a chemotherapy condition before the infusion. So this will be able to be done before the infection, so in a prophylactic way.
Yeah. Well, I don't know a lot about conditioning, but we are working on generating allogenic CAR T-cell therapy. So I think we are going to still be able to condition if the conditioning does not take a long period of time, which I don't know if anyone knows because it could be helpful, but I think we are still able to administer the therapy on time. And I don't even know even if it's an allogenic CAR T-cell therapy, the conditioning is still needed. Maybe no? So Natalia says no or yes.
You didn't need to make space
For the new to autologous. Okay, so there wouldn't be a need for conditioning. Therefore, I think we are pretty much safe to do it.
Okay, thank you for your talk.
Thank you.